Again, my name is Coughlin. I'm really excited to be here to be among such a great group of speakers. I won't be talking about preschoolers, but I will be talking about slightly older children. In particular, I'm going to be talking about the neural correlates of developmental differences in episodic memory flexibility. So episodic memory is our ability to form and retrieve memories of events that are spatially and temporally isolated and contextually rich. It's an inherently flexible ability, meaning that we can retrieve these memories across a variety of contexts. And this flexibility is really important as it supports our ability to succeed at everyday tasks, such as remembering where we parked our car when we're still in the office, to remembering some of our most personally meaningful and significant past experiences at any given moment. And because this ability is so critical to the human experience, it's really important that we understand how it develops. So research has shown improvements in episodic memory during childhood and adolescence, and there's large evidence that these improvements are supported by changes in cortical structure and function. However, there's also reason to suspect that changes in structure and function of the hippocampus may play an important role as well. For example, research has shown differences in the volume of different hippocampal subregions in children 8 to 11 compared to adults. And there's also some research suggesting that there might be functional differences, particularly in how the hippocampus builds a representation and then reinstates it in the service of episodic memory. So one of the main interests in our lab and focus of the study that I'll be presenting today is to further understand and characterize how these hippocampal differences in function may relate to improvements in episodic memory. And there's two possibilities that we are currently exploring. The first is that improvements in the precision of hippocampal presentations or representations may support the development of episodic memory. And this just means that with age, the hippocampus becomes better able at creating more precise representations. So in children, this would mean that when they go to retrieve a memory, they're better able to retrieving a specific memory and less likely to confuse it with similar memories. The second possibility that we're exploring, and let me say that these possibilities are not mutually exclusive, and there's many other possibilities, but the one that we're exploring, and that's the focus of the study today, is that improvements across development in episodic memory may be due to improvements in the ability of the hippocampus to flexibly encode and retrieve memories. So what do I mean by this? Well, ideally, whenever we experience an event, the hippocampus will bind the contextual features of that event into a mental representation that can be retrieved across varied contexts. And indeed, research suggests that it's actually more difficult to retrieve a memory when the context changes as opposed to when we're surrounded by contextual features that remind us of the original event. So, for example, having met me at the Flex conference today, it should be easier for you to recall who I am if you were to see me again at a future Flex conference, which would be great, than if you were to see me again in an entirely different context, such as at the beach or a sports stadium. And this is because a match between the encoding and retrieval context facilitates retrieval of the memory, whereas a mismatch makes it more difficult. Despite this, being able to retrieve memories across various contexts is an essential feature of episodic memory. It's a really important feature of it, and we refer to it as mnemonic flexibility. And there's evidence that the hippocampus may support improvements in this mnemonic flexibility, and that certain regions of the hippocampus in particular might support it. So, for example, research has suggested that there's a functional dissociation across the long axis of the hippocampus, such that interior regions may be more involved in the flexible encoding and retrieval of associations between events. Now, having said that, there are other studies that have operationalized flexibility differently, that have found patterns of association associated with flexibility that extend beyond the interior regions. But for the purpose of our study, we're focusing on flexibility in associative recognition, for which anterior regions have been implicated. So interestingly, there are there is support for age-related differences in the recruitment of the anterior regions of the hippocampus during the retrieval of episodic memories. And behaviorally, we also know that young children in particular really tend to benefit from a contextual match of event at encoding and retrieval. So together, these findings provide some support for the notion that age-related differences in hippocampal contribution to flexible encoding and retrieval of memories may actually partly support the development of episodic memory. And this is an idea that we're exploring in the study I'm presenting today. So in our sample, we're looking at 8 to 9-year-olds, 10 to 11-year-olds, and young adults. Unfortunately, our younger children, our child age groups are a little bit small. We're hoping to add a few participants to those groups. And we chose these age groups based on research showing differences in their ability to flexibly encode and retrieve memories. So I apologize that these pictures are a little small, but I'm going to talk you through the main fMRI task which we designed to address study aims. Specifically, participants completed 4 sessions during which they completed an encoding run followed by a retrieval run. During the encoding run, they were shown 2 pictures presented in a vertical orientation of everyday objects, and they were simply asked to report which of the 2 objects was heavier. And this question allowed them to process the co-occurrence of the pictures of objects while still maintaining separate representations of them. Next, they conducted a retrieval run during which, again, they saw 2 images presented of objects in a vertical orientation. And this time, they were simply asked to report whether or not they had seen this pair of objects together at encoding. Importantly, the pictures were presented in either the same location that they appeared at in encoding or in a different or flipped location than they appeared at encoding. And this represents our flexibility manipulation. So there are 5 different kinds of experimental conditions that were represented across the retrieval trials, and I'm going to briefly talk you through each of these. However, I want you to really pay attention to the first 2 because those are the results that I'm going to be focusing on today are from those first 2 conditions. I want to go through the other experimental conditions because they are an important part of the experimental design, and we are definitely going to be looking at them in future analyses. So, I hope that you can see this back here, but basically this is the old pair same location condition. And in this condition, participants were shown 2 pictures at retrieval together that were also shown together at encoding. And importantly, these pictures were shown at the same location in encoding as they were shown together in retrieval. So here, during encoding, they saw a Lego on top of a starfish paired together, and then again at retrieval, they saw the same pairing of a Lego and starfish, with the Lego again being above the starfish. So this would represent a contextual match between encoding and retrieval. The next condition was an old pair flip location condition. Again, in this condition, they saw 2 pictures paired together at retrieval that were also paired together at encoding. However, this time, the spatial location of the pictures was flipped between encoding and retrieval. So in this example, they saw a shovel and a violin paired together at encoding, with the shovel appearing above the violin. And at retrieval, they saw the same objects together, but the violin was now paired on top, of the shovel. So this condition represents a contextual mismatch between encoding and retrieval and would likely place higher demands on flexible encoding and retrieval. Now, for the next 3 conditions, participants were shown pairs of objects during retrieval that were not shown together during encoding. So in a new pair, same location condition, they were shown 2 pictures at encoding, but were seen separately at encoding. But in the same location at encoding as they were presented at retrieval. In a new pair flipped location, they saw 2 pictures paired together at retrieval, at encoding, and in this case, the position of the pictures at encoding was flipped so that they appeared in the opposite location during retrieval. And finally, there was a completely new pair condition such that participants were shown pairs of items at retrieval that included pictures that were not shown at all during encoding. So I'm going to briefly talk you through some behavioral results. This graph shows the performance rate across the different conditions I just described by age. The blue bars represent the old pairs, the green bars represent the new pairs, and the orange bar represents the completely new pair. The solid bars represent the same location condition where there was a match in the spatial location between encoding and retrieval, and the white bars represent the flip location condition where there was a mismatch between spatial location at encoding and retrieval. Results revealed an effective accuracy such that overall participants had higher hit rates compared to false alarms, and there was also a main effect of our flexibility condition, whether or not the pictures were presented in the same or flipped location. Now, although the age by flexibility condition interaction was not significant, if you look at these bars, you can see that really it was only the 8 to 9-year-olds who recognized the old pairs in the same location more frequently than they recognized the old pairs in the flipped location. This difference between conditions was not apparent in the older child age group nor the adults. And this suggests that there may be some improvements in flexible encoding and retrieval. Interestingly, although there was not a difference in the actual performance rates, the older children and the adult groups were still slower to respond in the old pair flip location condition than the old pair same location condition. So now I'm going to briefly talk you through some functional results, looking at whether differences in hippocampal activation may possibly relate to some of these observed differences. And we're going to focus on the hippocampal head in this analysis, specifically an anatomically defined ROI of the hippocampal head. Now, in our lab, we like to look at all regions of the hippocampus. But for the sake of brevity and because the interior region has been more heavily implicated in flexible encoding and retrieval, we're focusing on this region. To easily assess memory-related activation, we simply computed difference scores where we subtracted activation for misses from activation at hits to provide just an index of memory-related activation. And these difference scores were computed separately for the same versus the flip location conditions, as well as separately for activation at encoding versus retrieval. So consistent with our predictions, results revealed a significant age by flexibility condition interaction. And I'm graphing this interaction here. The blue bars represent the activation scores, different scores for the old pairs that were presented in the same location. And the red bars represent the activation difference scores for the old pairs that were represented in the flipped location. And as you can see, the hippocampal head was recruited more heavily for adults during flexible retrieval. And this was not observed in the two child age groups. And in fact, the youngest age group, the 8 to 9-year-olds, seem to recruit this region more heavily for the old pair same location condition, so when the context at encoding matched the context at retrieval. So together with the behavioral results, we feel that this finds some preliminary support for the notion that the development of episodic memory may be partly supported by age-related differences in hippocampal function and how it supports flexible encoding and retrieval. However, these results seem to imply that this pattern of activation was seen across both the encoding and retrieval phases of the study. And in fact, I want to mention that there was a trend for an interaction between age, flexibility condition, and encoding and retrieval, the mnemonic phase. So here I'm presenting a graph of the same activation, the same interaction I just showed you. Graphed separately for activation at encoding at the top and activation for retrieval at the bottom. And as you can see, the results I described just a minute ago are heavily driven by activation at retrieval. Indeed, at encoding, the effect of flexible retrieval that was observed in adults for activation of the hippocampal head was attenuated. See any difference in the youngest age group according to flexibility condition. Interestingly, during encoding, there really between activation for hits and misses. And if anything, there seemed to possibly be a slight advantage for misses in terms of perhaps a little bit more activation from items that were subsequently missed during retrieval. And we found this really puzzling and tried to think about what this might mean. Of course, there's a lot of different options and we're exploring them. But one is that, if you'll recall, during our encoding task, we asked children and adults to compare the two pictures and provide a weight judgment. Now, it's possible that during encoding, they were not only attending to the picture pair, but also something specific about the individual pictures and their context, such as the picture and its weight judgment, or the individual picture and where it actually appeared spatially on the screen. And luckily, after the fMRI task, we included a post-scan source task that took place right outside of the scanner, where we actually tested the association between individual items and their spatial context. Specifically, we showed items that were from the same location, old pair condition. We presented these items in the middle of the screen, and we asked children whether or not, and adults, whether or not they had seen these items at the top or the bottom of the screen. We thought that perhaps if they were attending more to an item specific association, an item context association, that performance on the source task might relate to the activation patterns that we were seeing. To do this, we simply did a correlation between encoding activation for items that were subsequently missed versus items that were subsequently remembered, and looked at the relationship between that activation and source task accuracy. And for the encoding activation for items that were subsequently missed, there was a positive correlation with the source task performance. So children and adults, performing better on the source task, which was looking at the association between items and their spatial context, actually related positively to more activation at encoding for items that were subsequently missed during retrieval. And this was not observed for the encoding activation of the old pairs that were subsequently remembered during retrieval. Now, this is still a possibility that we're actively exploring. The correlations aren't really significantly different, but we're finding this pattern across the different conditions. And so we find it interesting and something that we're going to continue to pursue. So in conclusion, encoding activation was not sensitive to our mnemonic flexibility manipulation, but it may be capturing the encoding of some other item context association. Interestingly, though, the retrieval activation did provide support for our hypothesis that age-related differences in hippocampal contribution to mnemonic flexibility may partly support episodic memory development. And there's a lot of data here, so we're still hoping to dig into these questions and have a little bit more clarity as we continue to analyze it. So I'd like to thank all of you for your attention, the wonderful families who participated in our research project, all the members, all the members, and my memories of my